Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study that uses cutting-edge genetic techniques to understand a debilitating muscle disease in cats. This research highlights how precision medicine, powered by whole genome sequencing, is revolutionizing our ability to diagnose and potentially treat genetic conditions in our feline companions. Our story begins with a young, 1-year-old, domestic short-hair cat who was brought to a veterinary specialty hospital with a perplexing set of symptoms. This wasn't just a case of a cat being a bit clumsy. This feline was experiencing progressive difficulty walking, a condition that had been worsening since it was 6 months old. Imagine a cat, once agile and graceful, now struggling with every step. But that wasn't all. The cat also had macroglossia, which is an abnormally large tongue, and dysphagia, or difficulty swallowing. The cat's tongue was so enlarged that it constantly protruded from its mouth, causing excessive drooling. On top of that, the cat's neck and shoulders were noticeably thickened and rigid, adding to its discomfort and limited movement. It was clear that something was seriously wrong, and the veterinarians suspected a congenital neuromuscular disease. To get to the bottom of this mystery, the veterinary team performed a thorough neurological examination and standard laboratory tests. They also took biopsies of the trapezius muscle, with the owner's consent, to examine the tissue at a microscopic level. The initial lab results revealed some significant red flags. The cat's aspartate aminotransferase, AST, and creatine kinase, CK, levels were sky-high. These enzymes are usually indicators of muscle damage, with the CK level being remarkably elevated. The cat also had mild hypokalemia, or low potassium, which can also point to muscle or nerve issues. Under anesthesia, small samples of muscle were taken from the cat's trapezius muscle. Some samples were preserved in formalin, while others were immediately frozen. The frozen samples were sent to a specialized neuromuscular lab at the University of California, San Diego. There, the samples were carefully analyzed using a range of histochemical stains and reactions to identify any abnormalities. The formalin-fixed samples underwent routine processing to prepare them for examination under a microscope. The lab team then used a technique called indirect immunofluorescent staining, applying various antibodies to highlight specific proteins associated with muscle structure and function. These antibodies were designed to bind to dystrophy-associated proteins, helping the researchers determine if any were missing or behaving abnormally. The immunostaining of the affected cat's muscle tissue was carefully compared to archived samples of healthy feline limb muscle to identify any differences. The muscle biopsies confirmed the veterinarian's fears. The cat was suffering from a degenerative and regenerative myopathy, a disease of the muscle. This meant that the muscle tissue was breaking down and attempting to repair itself, a hallmark of muscular dystrophy. To pinpoint the specific type of muscular dystrophy, the researchers used immunofluorescent staining to look for the presence or absence of key proteins associated with the condition. What they found was telling. The dystrophic muscle showed no signs of staining for dystrophin, a crucial protein that provides structural support to muscle fibers. Specifically, antibodies targeting the rod and the C-terminus of dystrophin failed to bind to the muscle tissue, indicating a deficiency in this protein. In contrast, staining for utrophin, a protein similar to dystrophin, was increased in the dystrophic muscle, suggesting the body was trying to compensate for the lack of dystrophin. The researchers also observed regenerating fibers, highlighted by staining for developmental myosin heavy chain, DMHC. Staining for laminin alpha-2 and collagen-6, two other proteins involved in muscle structure, appeared normal compared to control tissue. Based on these findings, the cat was diagnosed with a dystrophin-deficient form of X-linked muscular dystrophy. But the investigation didn't stop there. To understand the genetic basis of this cat's condition, the scientists turned to whole genome sequencing. DNA was extracted from the archived muscle tissue and sent to the University of Missouri for sequencing. The extracted DNA underwent rigorous quality and quantity checks using agarose gel electrophoresis to ensure it met the standards for sequencing. A sequencing library with an average insert size of 550 base pairs was constructed using the extracted DNA. This library was then pooled with around 21 other cat genomic sequencing libraries and loaded onto a NovaSeq 6000 for sequencing. This process generated approximately 30 times the sequencing coverage, ensuring high accuracy. The sequencing data was processed using a specialized Nextflow workflow, following the best practices outlined by the Genome Analysis Toolkit, GATK. The reads obtained from sequencing were mapped to the domestic cat reference genome using Minimap version 2. Duplicate reads were marked using Picard version 2 to avoid errors in variant calling. Specific tools from GATK 4.2 were used for genotyping, constructing variant databases, and hard filtering. The team then compared the cat's entire genetic code to a vast database of cat genomes, known as the 99 Lives Cat Genome Sequencing Dataset. This dataset includes whole genome sequencing data from 362 cats and whole exome sequencing data from 61 cats. By comparing the affected cat's genome to this extensive database, they were able to pinpoint unique genetic variants that might be responsible for the disease. The National Center for Biotechnology Information, NCBI, variant effect predictor was used to characterize the variants. The exonic variants and 10 base pairs flanking each exon were filtered and visualized using VAR-seq software. The researchers focused on variants that were unique to the affected cat and likely to disrupt gene function. After sifting through the data, they found a promising candidate, a stop-gain variant in the dystrophin DMD gene. This variant, called C.4849C/T, P/GLN1617-TER, essentially tells the cell to stop producing the dystrophin protein prematurely. The variant is located on the X chromosome at positions shown on the screen. Because this mutation occurs early in the gene, it would result in a truncated protein, missing a significant portion of its structure. The team predicted that this loss of function (LOF) variant was the likely cause of the cat's muscular dystrophy. The variant had good sequencing coverage with 12X sequencing reads, indicating reliable detection of the region on the X chromosome in the male cat. To confirm their findings, the researchers used Sanger sequencing, a more targeted method of DNA sequencing. PCR primers were designed using the cat reference assembly as shown on the screen. The PCR forward and reverse primer are listed on the screen. The PCR conditions were optimized for annealing temperature and MgCl2 concentration. They amplified the DMD gene fragment from the affected cat, an unaffected female sibling, and 2 control cats. The Sanger sequencing results were clear. The affected male cat was hemizygous for the nonsense LOF variant, meaning it had only one copy of the mutated gene on its single X chromosome. In contrast, the unaffected female sibling did not carry the DMD variant. Both control cats were hemizygous or homozygous for the normal, wild-type allele. This confirmed that the mutation was indeed present in the affected cat and absent in its healthy sibling, strengthening the link between the variant and the disease. This variant was unique to the affected cat within the 99 Lives dataset. With the genetic variant identified, the researchers turned to bioinformatics tools to predict its impact. They used resources at the NCBI website, including ClinVar, the Genome Data Viewer, BLAST, and the Genome Aggregation Database, NOM-AD, to compare the cat variant to similar mutations in humans. The MutPredLF web application was used to evaluate the effect of the LOF variant in both cats and humans. Multi-species sequence alignment of the exon containing the variant was performed using CLC Sequence Viewer 8. The NCBI cat reference assembly F.Cadis_FCA126 is annotated with 8 identified DMD transcripts in the domestic cat, which code for 78 exons and approximately 4,511 amino acids. The cat variant is positioned in exon 35, which has over 93% homology, with approximately 13 human DMD transcripts containing exon 35. The ClinVar database revealed that a similar mutation in humans had been observed in familial cases of Duchenne muscular dystrophy, DMD, and is considered pathogenic. Like the cat variant, the human mutation is a cytosine to thymine transition that changes the codon from CAA to TAA, creating a premature stop signal. This variant is not present in the NOM-AD population database, indicating its rarity. The MutPredLF application predicted a score of 0.46141 for the cat variant compared to a score of 0.45563 for the human variant. Scores above 0.7 are considered the most deleterious by this application. Multiple species alignments of the region across diverse mammals and zebrafish showed high conservation of both nucleotides and amino acids, further supporting the importance of this region for dystrophin function. This study not only identified a novel genetic variant responsible for X-linked muscular dystrophy in a cat, but also highlighted the power of precision medicine in veterinary practice. By combining clinical observations, advanced molecular techniques, and extensive genomic databases, the researchers were able to make a definitive diagnosis and gain insights into the underlying mechanisms of the disease. Muscular dystrophies are a group of genetic diseases characterized by progressive muscle degeneration and weakness. Duchenne muscular dystrophy, DMD, is the most common and severe form, caused by mutations in the dystrophin, DMD gene. This gene is one of the largest in the mammalian genome, and mutations within it can lead to various forms and severities of the disease. The ClinVar database has documented numerous variants in the DMD gene as likely pathogenic or pathogenic in humans, leading to different forms, severities, and presentations of the disease. Precise identification of the causal variant is crucial for prognosis, genetic counseling, and determining appropriate treatments. For example, read-through compounds such as atalurin are used for patients with nonsense, stop codon mutations, while exon-skipping treatments are also employed. Recently, delandestirgene moxeparvovec, an adeno-associated virus, AAV, vector-based gene therapy, was approved in the United States for treating ambulatory DMD pediatric patients with a defined variant in DMD. Ongoing preclinical trials in canine models support the development of these treatments. Defining DMD variants in cats, like the one identified in the study, supports the use of cats as a biomedical model in preclinical trials, similar to canine models. Feline X-linked MD was the first genetically characterized cat model for a human disease. The initial characterization involved a deletion in the muscle promoter of DMD, but the exact extent and positioning of the variant were not determined due to the limitations of the techniques used at the time. Since then, 2 additional DMD variants in cats have been characterized, both in Maine Coon cats. One study described a nonsense variant in exon 11 of the feline DMD gene, leading to the loss of most of the dystrophin protein. Another study identified a Missense variant associated with a milder, Becker-like form of MD. The DMD variant in the present case, C.4849C/T, is a loss-of-function variant caused by a nonsense mutation in exon 35. This variant is predicted to truncate approximately 65% of the dystrophin protein and was unique to the case in the 99 lives cohort. The fact that the female sibling did not have the variant suggests that the LOF variant was likely transmitted by the queen as a heterozygous carrier, or that the case cat represented a de novo mutation. The ClinVar database contains an entry for the human counterpart variant, which has the same predicted effect and segregates within a human family with DMD, supporting the pathogenicity of the cat variant based on comparative genetic criteria. Loss-of-function variants are generally considered among the most pathogenic mutations, but their effects can vary. While some LOF variants have no observable impact, others can cause significant disease. In this case, the variant was validated by high-coverage and high-quality sequencing reads, as well as direct Sanger sequencing. The accuracy of the cat dataset has improved due to robust sequencing technologies, consistent methods of variant identification, and improved genome assemblies and annotations. The high conservation of both nucleotide and amino acid sequences within the region across diverse mammalian species also supports the determination of pathogenicity for the variant. Without considering candidate genes, the recessive/hemizygous model identified 71 additional unique variants that could potentially be candidates for the disease. However, only 2 of these were within coding regions of genes, Missense variants in the sodium/potassium transporting ADPase subunit beta 3 gene, and an uncharacterized gene. The remaining genes, uncharacterized genes, and intergenic variants would require extensive experimentation to define causality. Similarly, most of the variants identified when considering the dominant model are non-coding, and most of the LOF frameshift variants are in uncharacterized genes. While the gene Titan, TTN, has been associated with MDs, the clinical, pathological, and histological data in this case supports a dystrophin deficiency, making further investigation of other genes less warranted. The young age of the affected male cat, along with the markedly increased CK activity, dystrophic phenotype in the muscle biopsy sample, and non-detectable staining for the rod and carboxy terminus of dystrophin with increased utrophin expression, is consistent with the diagnosis of X-linked MD. Muscle hypertrophy, a consistent finding in X-linked dystrophin-deficient cats, also occurs in Duchenne MD in humans and in the golden retriever model. This hypertrophy is considered true hypertrophy, not pseudohypertrophy related to fatty and connective tissue infiltration or hyperplasia. In conclusion, the expected LOF variant effect, population data, genetic data compared to humans, and amino acid conservation all support the identified variant as pathogenic and causing the clinical signs of X-linked MD in this cat. Precision/genomic medicine efforts for the domestic cat and in veterinary medicine increasingly support disease variant and animal model discovery, providing opportunities for targeted treatments for companion animals. This study underscores the importance of genetic testing in diagnosing and understanding inherited diseases in animals. As these technologies become more accessible, we can expect to see even more advances in veterinary medicine, leading to better diagnoses, treatments, and ultimately, improved quality of life for our beloved pets. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.